Hello dear students, hope you are doing well. As we have already studied about the structure of chromosome and the human chromosomes we studied, and about this video I'm going to explain about the structure of human DNA and also RNA. So I hope it works and it helps you. So there is a DNA. First of all, we should know that inside human body or inside human cell, there is something we call it genetic material, or we can call it nucleic acids. So normally we have DNA and RNA. So now I'm going to mention the structure of DNA. So simply DNA is the abbreviation of deoxyribonucleic acid. So why do we call it deoxy? Because there is one oxygen missing in the pentose sugar. Later on we are going to explain. First of all, in 1953, there were biology scientists, they studied and let's say they found DNA, we can say that. And there were two scientists. Their name was, let's say, Watson and Crick. But the idea for DNA structure, it basically consists of two anti-parallel, let's say, chains. This is very important to know that. Why do we call anti-parallel chains? Look, in here, normally parallel, unlike this, two lines, they go straight. But when we talk about DNA, as we have seen, you see the lines. They are anti-parallel. So they are cutting each other. And there are some meeting points here. That's why we call anti-parallel. Okay. And you should know that inside here, it says when in the shape of double spherical and nitrogen bases, for one of the chains connect with the base which relate to the complementary in the opposite side. So when there is a line, there is a side, opposite also there is complementary. What do you say the meaning of complementary? We have nitrogen bases that we are going to explain later in detail. You see, they are the complementary of each other and there is a bond between them. Okay, and also a sugar circle and phosphate group in each the chain, they are connected by covalent bonds. So this covalent bond, it's a bond between sugar circles and phosphate groups. In each side, they have been connected to each other. This is very important. And normally, as we mentioned, DNA is the abbreviation of deoxyribonucleic acid. So regard it as the complex chemical compound. It represents in all living organisms and it is important for life. Why do you say it's important? Because we can say that it works like a memory. Transfers all informations from parents to the offsprings, generation by generations. So it represents the main nucleus or nucleus within the chromosomes, which forms from the chromatin reticulum. Do you remember we said inside the nucleus of this is the, let's say, cell membrane, and inside the cell we have nucleus, in eukaryotic cell I mean. So inside here, inside the nucleus, normally we have nucleoplasm and let's say nucleolus. So we are talking about the nucleoplasm. Inside here there is a liquid and we can find chromatin network, as we already mentioned that one. So inside the chromatin network, of course, we have chromosomes, and above the chromosomes, if you have this chromosome, we see the chromosomes are made up of two, let's say, sister chromatids, and each chromosome or sister chromatid has these DNA strand which is let's say, all located above the chromosomes. So in here, what do we have? Normally it says, and the chromatin material forms the units of nucleoplasm, as we said that. And you should know that there's something important in here, and I've told you already. DNA also represents in some cytoplasmic organelles. What does that mean? We said we have two organelles inside the eukaryotic cell. Also, they have their own DNA. This is very important. First of all, we have mitochondria. Secondly, we have plastids. Okay, now let's go in details. Normally, if we study the structure of DNA, normally we have three main chemical structures inside DNA. First of all, says pentose sugar. Okay, means sugar, carbohydrates. Okay, so it means that sugar can be found inside the structure of DNA. So it's called, let's say, deoxyribose. So the molecule of the formula of the sugar is what? C5H10 and O4. Normally it should be O5. You should know that. But it's not O5, isn't it? That's why we call it deoxy. Means one oxygen is missing. Okay, the name came from that one, deoxyribonucleic acid. Second group is phosphate groups. So these consist of an atom of the phosphate P, which connected by 4 atoms of oxygen. You see? 4 atoms of oxygen, not 5. And the alternate molecules of the phosphate and sugar form what? 2 sides of DNA chain. From each side we have sugar group and also phosphate. In other side, vice versa is true. So it means that, you should know that, each group of let's say phosphate group is connected with a group of sugar. By what? By covalent bond, which is combines between the sugar of one of the let's say nucleotides and phosphate group from other nucleotides. Please pay attention. So in here, if we have, let's say, a phosphate group, opposite will be sugar. In here, if we have sugar, there will be phosphate. And they have been connected by covalent bond. It's a chemical bond between phosphate and let's say sugar. And covalent bond is units 2 atoms as a result of cooperation of each of one of them by electrons. It's mostly related to the chemistry, so we are not going in detail. Last one, we have nitrogen bases. This is very important, nitrogen bases. Normally, it is a ring compound which contain nitrogen. So in addition to carbon and hydrogen and oxygen, except adenine. Please pay attention. This exception is important. Can be asked in filling blanks. I can ask you, all nucleotides on nitrogen bases has these atoms, these are, let's say, molecules, except adenine, which does not contain oxygen. That's why we call deoxyribonucleic acid. So the distance between each base to another is what? Is constant. It doesn't change. This is very important. Always, it doesn't change the distance between the molecules. Okay. Normally, we have 5 types of nitrogen bases, or we can call it, nitrogen bases, as we have here. Please pay attention. What do we have? We said we have 4, of course, but in each, let's say, the genetic material which we have DNA and RNA, there is a difference. You should know that. First of all, we can, let's say, divide them into 2 groups. Group 1, we call it pyrimidine, which includes all the following bases. We have thymine, we have cytosine, we have uracil. Please pay attention. Thymine, it is available in DNA only, okay? And in here, uracil only can be found in RNA. Here is the main difference between them. Always when we see the nitrogen bases, thymine can be found in DNA, which is not inside RNA, and uracil can be found inside RNA, which is not present in DNA. And cytosine available in both. Okay. Here is this group is about pyrimidine. But purine, the second group, is what includes of these bases, adenine and guanine. And they are both available in DNA and RNA. You should know that. So the linkage of the bases between 2 chains is not random, but it is, let's say, bounded. Yani, it means, so adenine in one of the chains always connect with thymine in another chain by 2 hydrogen bonds. Please pay attention, yani. In here, always adenine will be connected with the thymine by 2 hydrogen bonds. Where is it? In here. You see? This example. The example 1. So always adenine will be connected with the thymine, and the hydrogen bond, we have only 2 bonds between them. But the second example, let's say cytosine, always connects with the guanine by 3 hydrogen bonds in here. Group 2, you see? We have 3 hydrogen bonds between, let's say, cytosine and guanine. So it means that it's impossible for guanine to make a bond with adenine because their structure is different. Always cytosine and guanine should make 3 bonds, and adenine and thymine should make 2 bonds, double bond, we say. That's why we call them complementary base pairs. So for this name also is important, can be asked in fill in blanks maybe. They are going to ask you these examples, these nitrogen bases, which we call cytosine and guanine as an example. We call them complementary base pairs. So the order of the, let's say, bases in one chain of DNA molecule or RNA is complementing for the structure of the bases to the opposite chain. It means that always when there is guanine, there should be opposite guanine is C. Always in here, if we have thymine, there should be what? Adenine, isn't it? This is important. And in Muzari, how they are going to ask you? For example, it tells you there's an example, a nitrogen base pair, which a gene in one of the pairs is A, G, C, let's say T. So they will give you that one. So when an exam gives you this gene, asks you about write down the complementary base pairs, nitrogen bases for that one. What you are going to do? It's very easy. We said always adenine make 2 bonds, hydrogen bonds with thymine, isn't it? And guanine, we said, make a bond with cytosine. Cytosine make a bond with guanine. So thymine make a bond with adenine. This is the solution of any questions that can be asked in Uzari. Okay. There's something else says complementary base pairs are important in DNA's structure and function for 2 reasons. Also, there can be a Uzari question. They can ask you why it's important for 2 reasons. First, it says because the bonds between the base pairs help to hold the 2 DNA chains, of course. Just imagine if you have these 2 chains like this, there will not be any connection between them. Always, let's say it will be separated and it won't stabilize. It won't be stabilized. And the second reason says because of the complementary nature of DNA helps to explain how it is replication before the cell division. It's very important. So an example says if you know this, let's say, succession of the bases of one DNA chain, which is in here, so the question says complementary base pairs will be the opposite chain. Here is the solution, as I told you just now. If you have these question, the answer will be like this, which is thymine will be adenine, cytosine will be guanine, thymine again will be it should be adenine, sorry. Okay, it's adenine. It's a mistaken, like this. And the above shows DNA is very important because it is the forming material of genes and succession of the nitrogenous bases, which is determines the traits of organism. So for next video, for next lesson, I'm going to explain the structure of RNA. Thanks so much.